Welcome to this presentation on paper-based nanokit for amplification of HPV-16 or 18E6 oncoprotein detection for the screening of cervical cancer. My name is Lucy Mudhoni Mwai. I am from Kenya, and I want to take this opportunity to thank Beyond Sciences for giving me an international audience to make this presentation. Cervical cancer is usually caused by persistent sexually transmitted infection with high risk, HPV-16 and 18, around 70% of all the cases. It's the fourth female malignancy worldwide and second in low and middle income countries. And in Kenya annually we have around 5,250 new cases with 3,286 mortalities. There is actually need to improve screening for cervical cancer because there is late diagnosis as it is the case for most cancers, and screening is about 3.5% annually. This is attributed to lack of access to screening, lack of government initiatives among others, and challenges with common tests such as pap smears taking like 3 days, the cost. And now in this study we'll be using a biomarker called E6. It's an early oncogenic protein. It usually degrades p53, tumor suppressor protein, and therefore the cells are not able to conduct DNA repair, causing chromosomal instability, hence multiplication of cells and eventually cancer. Because it's present in pre-cervical cancer lesions and in cervical cancer, it is hence a main protein biomarker, and we can see that usually it's produced with others E1, E2 and E7, and it's early because we have also late L1, L2 and E4 also being produced at late expression. Our objective will be to amplify HPV-16 or 18E6 oncoprotein detection using a paper-based nanokit. And the first objective will be to develop a bioconjugate of now the gold nanoparticles with an antibody against HPV-16 and HPV-18E6 that is also conjugated to HRP. This is going to help us in the detection. Then to evaluate immune sensing mechanism of that conjugated antibody for now the HPV-E6 protein, and finally determine diagnostic accuracy of the nanokit using a gold standard. So this is our bioconjugation objective, our materials and reagents. Then we have now the first step of the noncovalent bioconjugation of setting experimental conditions. And the first thing we did was to determine the pH of the HRP antibody, which we got at 7.05. And then we need to adjust our colloidal gold solution to a pH 0.5 higher, and you can see now this is 7.83 compared to the antibody using borate buffer. And then we also determine the lambda maximum for the colloidal gold solution using UV spectroscopy before we now conjugate, and we obtained the lambda max at 523 nanometers. Now we perform the conjugation optimization protocol. We have to have different dilutions of our antibodies, and these are the various dilutions. Once we have had the various dilutions, we will take 900 microliters of each of the dilutions, and to each we add 100 microliters of gold solution, incubate for 2 hours and obtain the absorption spectra. Now this is now the spectra of the gold nanoparticles before conjugation, which we have said was at 523, and from our purchase at Sigma Aldrich, this 20 nanometer optical density of 1 gold nanoparticles was supposed to have a lambda max of 518 to 524, and therefore this lies within the limit. Now after we have conjugated, this is what we expect, that our gold nanoparticles will conjugate with antibodies, the antibody HPV-16 18E6 antibody. And now the conjugation in this case is passive adsorption, usually involves electrostatic and hydrophobic interactions, meaning it is not covalent, it is a non-covalent. It is easier because we are not having any chemical changes during the bioconjugation. Now, once there is conjugation, we usually have what we call a redshift, because the antibody replaces the negative charge citrate capping. Remember that our antibodies were citrate stabilized, and because of this, the localized surface plasmon resonance spectrum redshifts by a few nanometers, but it does not go beyond 400 to 900. Now, this redshift experienced in our experiment was 0.5 nanometers, as we can see in this diagram. So this is our shift from 523 to 523.5, and these ones represent the other spectra of the other dilutions, and we can see that none of them has had a redshift or an increase in the nanometers. Now we perform the dot blotch molecular technique, and this is going to help us to be able now to test our conjugated antibody if it's able to detect our E6 protein. So that is the very first part of this objective. Does the conjugate possess immune sensing properties? And then what is the lowest detectable amount? And finally, does this immune sensing property of E6 vary between a conjugated antibody and the unconjugated antibody? So this is the procedure where we first had now the differing dilutions. It's always important to have different dilutions of the protein, and this way you are able to assess how the reaction is. This is a chromogenic reaction, meaning colorimetric. You're able to have color because of the HRP that was conjugated to the antibody, and we are going to use a reagent called TMB. So first we spot our diluted concentrations on the nitrocellulose paper, then we block with BSA, and then we have now our conjugate antibody, then we incubate, then wash the membrane 3 times with TBS, T-buffer, then we add the TMB reagent. This will enhance color, and then once we observe, we'll have the color increasing intensity, of course, from the lowest dilution to the highest. So the one that is 0.02 milligrams per ml will have a higher concentration of the E6 protein, and therefore we expect a higher color development. And this is also going now to help us, the zero dilutions, in detecting the lowest detectable limit. Now this is the result. Now we had 11 samples and the negative control, and we can see that there is an increase in signal from 1 to 8, because from 1 to 8 we have an increasing concentration of E6. But of course, from this there is also some irregularities you can see here, and this is possibly because of bioconjugate instabilities. There is a possibility that bioconjugation was not very stable. Remember, it's non-covalent, and therefore that could be what is causing the challenge there. Now, limit of detection. Now from our silo dilution, you can see that the lowest concentration obtained, or where we have detection, is at this point, and this is 0.005 picograms per ml. And from this we can actually get a color code, where now, because we know these concentrations, if a patient gets this, then we know that the E6 concentration is high, and therefore they have progressed quite further into cervical cancer. Then signal amplification. This is important. This is our unconjugated antibody, and this is conjugated antibody. So we just want to see if we did not have the gold nanoparticles, our antibody would still detect our E6 protein, but it is more faint as compared to if we conjugate. So the gold nanoparticles actually help us to amplify. They have light properties that help us to amplify. And finally, now we can see that it's able to detect our E6 protein. Now, what of now comparing it with another standard test that is being used, which we call histopathology test, that is done. Now, here we first had to select our samples. We utilized cervical tissue, formalin fixed, paraffin embedded. We call them FFPE biopsy samples that had confirmed pathology from the University of Nairobi Histology Department repository. So you can see these are archived samples, and we did this because this is an exploratory test or a proof of concept study. So we just want to see how it's comparing, then we can have the study scaled up to, you know, a little bit more samples. So we recruited in this retrospective cross-sectional study women who are 18 years and above, diagnosed either negative or with precancerous lesions or with cancer. And we had these samples, we only used those that were 10 years. From the investigation, they haven't been archived for more than 10 years. We took those that were between 2012 to 2014. And we also chose bigger tissue samples where possible to ensure that we get maximum E6. We excluded women with other type of malignancies, or they had some form of treatment for cervical cancer, or those that had undergone any form of surgery. And these were the samples we obtained. We had a sample population of 50 samples. So now we need to do a protein extraction. We used lysis method. So we first have to homogenize the sample because it has been stored. So we do microtomy to extract our tissue. We deparaffinize to remove the paraffin that is used using xylene. Then we pellet, we deparaffinize again to ensure we remove all the paraffin wax. Then we rehydrate in ethanol, grades of ethanol 100, 90 and 70. Then we resuspend now in our lysis buffer with protease inhibitors to avoid it digesting our protein. Then we vortex on ice to preserve our proteins again. We centrifuge to remove any unsolubilized material, and then we collect the supernatant in fresh tubes. And this is where now our E6 protein is. Now, after we have done that, remember that we had said that we had normal cervical biopsies, we had mild dysplasia, we had CIN3, we had CIN4, microinvasive and macroinvasive. So this is now A, and we can see that in this reaction we did we had 10 samples. We can see that there was a false positive there. Essentially this should all be negative because this is a normal cervical biopsy. And then we have B, where we have mild dysplasia, and we can see the reaction is a bit faint. But then from these 4 samples we can see that there was a negative, which is a false negative, because essentially there should be a reaction. Then at C we have CIN3. We can see that there is improved detection as E6 is increasing. Then D we have CIN4. Again you can see we are increasing. And now we have microinvasive reaction, have 5 samples, and macro. Now this is full-blown cervical cancer, and we can see that the concentration is quite high. Now from this we draw a 2x2 contingency table that determines the parameters of diagnostic test quality. TP stands for true positive, false negative, true negative. And now we have put all the samples, and we said our samples were 50. Now from there, then we determine now the parameters of sensitivity, specificity, the predictive values and likelihood ratios. And we can see that from the lower and upper limit, our obtained values fall within the limits. You can see they fall within the limit. Now from there we extract something we call an ROC curve. ROC stands for receiver operating characteristics curve, and this one usually you plot sensitivity against 1 minus specificity to display the trade-offs between now specificity and sensitivity. And now when we plot this graph, we can see that the graph shows a consistent plot on the left side here and on the top, and this is a good curve. Normally after we have the curve, we determine the area under the curve, the AROC, and in this case we obtain 0.93875. The lower limit is usually 0.88 and around 0.99 as the upper limit, and you can see that ours falls very well within the area under the curve. Now what this area under the curve confirms is that our nanokit has a high discriminative ability. You know, we need to identify the people who are sick from those that are healthy so that we avoid unnecessary tests and treatment. So what do we conclude from this study? That it was possible first to bioconjugate the antibodies that are against the E6 oncoprotein with 20 nanometer citrate stabilized gold nanoparticles using passive adsorption. And then our conjugate possesses immune sensing mechanism for HPV 16 and 18 early E6 oncoprotein that is produced during early stages, and this is very good because it means it can be used very good for screening. The study also demonstrated that the conjugation with gold nanoparticles amplifies or improves the detection of E6. The study also gave us a commendable diagnostic accuracy when we compared it to the gold standard test. Now, our research outputs, we have been able to have 2 publications in 2020, and we also have a patent in Kenya. Now, the limitations, as we had seen earlier, is there is possible bioconjugate instability because of passive adsorption. This is our passive adsorption. Now, we need to consider having covalent. Let's consider have covalent bioconjugation in the subsequent test to ensure that the reproducibility issue is improved. Then we also need to have them tested on freshly collected cervicovaginal samples. You see, we used archived samples, so we need fresh samples. Then we need also more optimization to compare and ensure that the reaction was actually because of E6 combination with the antibody, so that we can eliminate any other microorganisms that could cross-react. So there is a lot to be done in this study. This is just a proof of concept or exploratory study, and I thank you so much for being part of this presentation.